Well, our next speaker is Miss Janet Chappell. She grew up in Lexington, Kentucky, and received her undergraduate degree from the University of Kentucky. While studying biology as a major, she happened to take an elective course in agroecology and has been hooked on to agroecology ever since. She is currently pursuing a master's degree in the Department of Soil Science at NC State University in Raleigh, North Carolina. And she and her advisor, Dr. Franz Luber, have been working on greenhouse gas reductions, measuring greenhouse gas emissions in agroforestry systems. So today she is going to talk about her work. So please join me in welcoming Janet.
Thank you to the Center for Agroforestry for extending the invite for me to present. I'm sorry my advisor, Alan Franz Lubers, can't be here because he would be such a great addition to this symposium, and this is right up his alley. So I'm here, and I'm going to present some preliminary results from my current master's project at North Carolina. Okay, so for the last year, we've been investigating how agroforestry systems impact greenhouse gas emissions. So conventional agriculture is the major contributor for anthropogenic greenhouse gas emissions, and agricultural soils actually account for 69% of nitrous oxide emissions, which is the largest source of N2O emissions. Carbon dioxide emissions from soil are also the second largest input of CO2 into our atmosphere. So it's really important that we consider alternative agricultural methods to reduce greenhouse gas emissions and sequester carbon. So agricultural systems can do this in two ways. You can reduce gas emissions and sequester carbon in soil and above ground biomass, and agroforestry is one alternative management system that may be able to do both of these. So our overall research goal was to provide a more complete process-oriented description of how agroforestry systems might reduce greenhouse gas emissions and sequester carbon. And our specific objectives were to assess the impact of agroforestry management on greenhouse gas emissions, nitrous oxide and carbon dioxide, as well as agroforestry's potential to sequester carbon and nitrogen fractions in the soil. Our general hypotheses were that greenhouse gas emissions would differ spatially and temporally in our agroforestry system, and that these emissions would vary with season due to environmental impacts such as temperature and moisture, and also that soil type would regulate gas emissions as well. The agroforestry site that I've been working on is located on the coastal plain of North Carolina. It's about an hour from Raleigh, and it's part of the Center for Environmental Farming Systems, which is a larger research unit that has many different sustainable and alternative agricultural projects going on, and the agroforestry system is one component of that. The study site is 7 hectares of alluvial river bottom, and it's located near the Neuse River. You can't see it, but just outside this picture is the Neuse River. You can see that there are tree species planted in staggered rows, and then in the alleys are cropped. In the past, it was a corn-soybean rotation. When I started 2 years ago, annual ryegrass was planted, and then this past summer, it was transitioned into warm season grasses. This is a picture of standing in the middle of one of the cropped alleys soon after ryegrass was planted, and it just shows you the 3 different tree species here are planted. We have longleaf pine, loblolly pine, and a cherry bark oak. This is an aerial view again, but I'm going to point out where I measured gas emissions. So it was in this zone right here. Because the agroforestry site is located near the Neuse River and it slopes downward, we have a gradient in soil textures. And I thought that it would be interesting to look at how differences in clay concentration and soil organic carbon and fertility influenced soil gas emissions. So I placed chambers in these areas in order to capture differences in soil properties. I had a total of 45 chambers, and so chambers were placed underneath the trees, in the middle of the cropped alleys, and then in the space between the trees and the cropped alleys. So I think someone yesterday told me that could be referred to as the tree-crop interface. And then here are some pictures of the gas chambers underneath the trees, and then you can see staggered in the cropped alleys. Our gas chambers consisted of aluminum steam tabletop pans, and what we did was we had the bottoms of these pans cut out, and that was inserted into the ground and left there permanently underneath the tree rows. I had to remove the bases when the site was tilled, planted, or fertilized. Then we had a top here that we fabricated with a little sampling port, and I collected gas samples with a basic polypropylene syringe and put those in glass vials and then transported them back to the lab to be run on a gas chromatograph. Soils, we also collected soil samples for analysis. We collected soil samples 4 times throughout the course of the year. From all positions where chambers were located, so in the cropped alleys, under the trees, and in that crop tree interface. These are some of the analysis we've done on the soil samples, but for the purpose of today's talk, I'm just going to focus on clay concentration, organic carbon and nitrogen, and microbial biomass carbon. So now I'm going to present some of our gas emission data, and I'm going to start with carbon dioxide as an introduction. You can see here that I've shown soil temperature, and this has been measured about a mile from my study site over the course of a year. I'm showing soil temperature because that is one of the main regulators of carbon dioxide emission from soil. And then I've overlaying here crop CO2 emissions. So each one of these points represents the average crop emissions from one sampling event. And you can see we saw a temperature-related response to CO2 emissions with higher emissions in the spring and summer months compared to the winter months. But you can also see we saw some dips in CO2 emissions. And those points right there corresponded to low soil moisture and times when there was not much rain. And then again, these peaks up here are warmer temperatures, but also when there were more heavy rainfall events before I went out and sampled. So I went out and sampled many times, but this is just 42 of the sampling events that we used in our data analysis. So this is from the cropped area, and then this, the little green triangles here connected, are from our CO2 emissions from the tree rows. And in all cases, carbon dioxide from the tree rows was less than the cropped alleys. You can see it also follows a seasonal pattern corresponding with increases in temperature. Next is seasonal changes in N2O flux. I've shown right here, this is rainfall throughout the course of the year because nitrous oxide emissions are often strongly correlated with soil moisture, and you can see peaks after rainfall events. So that's what we were expecting to see as well. And so again, brown dots are nitrous oxide from the cropped alleys, and we did observe some peaks after rain events, but it was not consistent. We did, however, observe a peak after addition of nitrogen fertilizer, but that peak was temporary and came back down to base levels a week after. I did notice that after we applied the nitrogen fertilizer, I would get some what I call like hot spots of nitrous oxide production. So it appears that the addition of the fertilizer did stimulate nitrous oxide production because nitrate is a substrate for denitrification. And then again, here is the nitrous oxide emissions from under the tree rows. And in most cases, it was lower than the cropped fields, the cropped alleys, excuse me. And we did not see any peaks in nitrous oxide after fertilizer addition or after many heavy rainfall events. So, you know, it's possible and likely that the trees were absorbing any excess water or nutrients, and therefore we didn't see any spikes due to denitrification. So to further investigate how environmental conditions affected gas emissions, we split our soil sampling dates into 9 categories based on volumetric water content and soil temperature. And you can see here I've listed the ranges for low, medium, and high. This will come into play a little bit later when I show you some graphs. So this is how my sampling events ended up being distributed into those 9 categories. The bulk, right here, well, not the bulk, but a good portion of sampling events was when soil temperature was low and soil moisture was high. So this is in the winter time and in the early spring when there was a lot of rain too. And I want to let you know that this category also includes the dates soon after the site was fertilized. So we wanted to investigate what was the influence of soil moisture and temperature on respiration, and this is what we expected to see was that as temperature increased, soil respiration would also increase once a certain soil moisture threshold was reached. So you can see this is what we wanted to see or what we expected to see. And now I'm going to show you what we actually saw. So here on the x-axis is soil temperature, and then on the y-axis is CO2 emissions from the cropped alleys. This is data from the low soil moisture sampling dates, and next to each point here is days after planting. I've included days after planting for these carbon dioxide graphs because tillage, planting, disturbing the soil will increase the exposure of any crop residue to microbial attack, which will, when heterotrophic microorganisms are decomposing that plant material, they're going to release carbon dioxide. And so we would expect that less days after planting to see a higher flux in carbon dioxide. So this is low soil moisture. This is medium soil moisture here, and we see a nice increase of soil temperature, but this is hard really to explain. And I think what you'll see is that gas fluxes can be very variable, and it's a complicated story, and it's not always easy to explain or predict, which is why we still need to go out and measure what's going on in the field. So this is high soil moisture category, and we do see an increase in gas emissions with an increase in temperature and less days from planting. And that was from the cropped alleys, and this is from the tree rows. I didn't include days after planting because the tree rows were not disturbed from any kind of activity. So low moisture, medium moisture categories, and the higher moisture categories, and we do see a nice increase of predictable, more so, soil temperature. And you'll notice that, again, on this graph, the y-axis only goes up to 12 kilograms of carbon dioxide per hectare per day, where as in the other graph, it's higher. So in all cases, for crops, so in all cases, the trees emitted less carbon dioxide than the cropped alleys. We also did this analysis for nitrous oxide emissions in the cropped rows in the trees, and in here, these numbers correspond to days after the fertilizer event. So this is low soil moisture, medium soil moisture, and high moisture. So, we expected to see an increase with that. This right here, it looks funky, but it can be explained by, even though this number is high days after fertilizer event, it actually includes many sampling dates that were right after a fertilizer event and many days after a fertilizer event. So the sampling dates soon after the fertilizer was applied, we saw exponential increases in nitrous oxide production, and that's what's contributing to this high level here. And then for tree rows, again, nitrous oxide emissions are low. And I've included the days after a fertilizer event on this graph too, because we think that, I've hypothesized that maybe the trees can absorb any runoff of nitrate from the cropped alleys. So maybe some fertilizer application had some effect. So the trees, we don't see too much of an increase with soil temperature at the medium and high moisture levels. You do at the low soil moisture. So there maybe seems to be an interaction, but we still need to look into that as well. So I also wanted to look at how soil texture may influence nitrous oxide emissions. And here I've graphed soil clay concentration and N2O emissions from the cropped alley and the tree row. And we did see that nitrous oxide emissions correlated with clay concentration only in the cropped alleys, not in the tree rows. So I find that interesting, and maybe the trees are creating some kind of microclimate, some kind of other soil properties that are involved there that are influenced by the trees. So we wanted to investigate why that was. So first I looked at soil organic carbon, and this is from the 0 to 20 centimeter depth, and it's values just from my first sampling date. And then I've plotted here clay concentration on the x-axis, and soil organic carbon increased with increasing clay concentration up to about 200 grams per kilogram of clay in the soil, and then from there it kind of plateaued out. And in the field you can actually see how the vegetation in the higher clay concentrations is much more robust. The trees are taller, the grasses grow better. So we can see that too in the field. And why soil organic carbon? Well, because it is a substrate for microorganisms and a major factor in regulating gas emissions. We also looked at microbial biomass carbon and clay concentration, and as clay concentration increased, we also saw an increase in microbial biomass. In both of these cases, you can see that there doesn't seem to be a clear difference between cropped alley and tree rows for these forms of carbon. So that may not be what's explaining the difference between gas emissions from the cropped alleys and the tree rows, but it may help explain why we see increases in gas fluxes from the cropped alleys with increasing clay concentration. This last graph shows clay concentration and soil moisture. So the soil with the higher clay with the finer textures have more pore size have a greater pore size distribution than do the sandier soils, and so they're able to hold more water and may be more easily able to support some anaerobic conditions which stimulate denitrification. So that may be why the soil moisture, while we're seeing greater increases in nitrous oxide at those higher clay concentrations. But once again, the difference between the trees and the crops for soil moisture is not too much different. And so what's explaining this difference between crops and trees for gas emissions, and that trees appear to, the tree rows, and there's not as much gas emissions. And so we hypothesize that there is some kind of microclimate effect going on, whether maybe the tree roots aren't respiring as much as older tree stands. So our trees are younger, they're about 7 years old. So root respiration may not be as high as in other studies that measure soil respiration from trees to be greater than cropped alleys. And the trees may just be better at absorbing any kind of excess nutrients and supporting better soil moisture conditions that don't lead to anaerobic conditions. So in summary, we found that gas emissions did vary with space and time, and that proximity to tree row did affect gas emissions. And I think it's just important to know that these things aren't always predictable, and therefore it's still important for us to go out and assess how systems like agroforestry management may impact these gas emissions. We did see typical peaks associated with fertilizer application, and we did note that nitrous oxide fluxes were correlated with soil clay in the cropped alleys, but not in the tree rows. So our results indicate that to some degree, agroforestry management practices will influence the microclimate, which may affect the magnitude of soil respiration and nitrous oxide emissions. And tree canopies may also be keeping soil drier and buffering temperature, and that these systems may be suitable for marginal agricultural lands where where soil is not as fertile and profitable. So I would like to acknowledge my advisor, Alan Franzlugers, and my co-advisor, Dr. Weishi. Both are soil scientists at North Carolina State University. Dr. Franzlugers is also a USDA ARS soil ecologist. My committee members, I am part of a wonderful lab which is so supportive, and our funding from the Forest Service and the Center for Environmental Farming Systems as well. So, any questions?
All right, thank you, Janet. And we do have time for questions.
You said you put in warm season grasses. What type did you put in?
It was a mixture of 3 grasses, and I cannot remember off the top of my head exactly what that mixture was. Sorry.
Is this working? Yeah. I was just curious about reuptake of released CO2 in the canopy of the trees, whereas your crop wouldn't necessarily have a canopy to recapture that released CO2. And have you looked into that at all?
No, I haven't. I was just measuring soil respiration on the soil surface, and I think it's important to note that there's no way I can partition between root respiration and microbial activity that leads to carbon dioxide emissions. So, no, I have not looked at any differences in gas diffusion up to the soil, but that would be something to consider.
How wide was your the alley crops between? How was your study configured? How wide was the crop area? How wide was the spacing on the trees? Was it single row, double row, or just basically house sides? And did you ever think about putting soil moisture, temperature type situations in the tree rows to see if the amount of temperature is cooler in that soils than it was on the outside?
Okay, great. So the cropped alleys that I measured gas emissions in were 12 meters wide. And then the tree rows consisted of actually 3 staggered rows of trees planted. And just some more details, we actually have the 3 different tree species. So I measured gas emissions from each of those different tree species. I put 3 chambers under each grouping of tree species, but I repeated that 3 times. And then I did measure soil temperature and soil moisture alongside each chamber every time I went out and gas sampled, and I did notice that there was lower soil temperature under the tree rows than in the cropped alleys. So there was a position effect without a doubt. Same for soil moisture.